Welcome to the second half of today's session on Introduction to Aging. It's a great pleasure that I have a chance to introduce Dr. John Papaconstantino. I've known Dr. Papaconstantino for a long time, and I worked with the Pepper Center and as our university expert on molecular aspects of aging. And Dr. Papaconstantino received his PhD in biochemistry at the Johns Hopkins University in 1958 and postdoctoral studies at Carnegie Institute of Washington Department of Embryology in 58 and 1960. He served as a senior research staff in biology division at Oak Ridge National Laboratory and is presently a professor in the Department of Human Biological Chemistry and Genetics at UTMB. He's also the Bertha and Robert Bakash Distinguished Research Professor of Aging and the Mary and J. Palmer Saunders Professor for Excellence in Teaching at UTMB. And I know as a mentor, he's extremely wonderful with students, and his passion for what he does in his research and to teach is, there's hardly anyone better that I know. His research focuses upon the biology and genetics of longevity determination in vertebrates, and his goal is to utilize different techniques of functional genomics and proteomics to understand the molecular genetic basis of longevity. He's going to be talking to us about the molecular basis of aging and longevity.
Thanks, Tony. That's a very nice introduction. Thank you. Let's see. So I need to turn this on first. Okay, got it. Okay, so as the title says, what I would like to do is present you with an overall view and understanding of what we know so far about aging with respect to the genetic factors that determine aging and longevity, the environmental factors that affect aging and longevity, and indeed what you'll see is that the genetic factors and environmental factors interact to actually determine the rates at which we age and the rates at which we develop age-associated characteristics. There's a lot of molecular biology and molecular genetics going on now with respect to trying to understand the biological basis of aging, and I'm going to be presenting you with some of the latest studies that implicate both the fact that indeed our longevity and rates of aging are genetically determined, and that environmental factors play an important part in how rapidly we age or what our lifespan will be. So let's get started. Okay, well, as you can see in this beginning, I'm asking and making the statement that aging in higher organisms, well, aging in general, is a complex biological process that occurs in all organisms. And I'm going to show you that indeed aging occurs in yeast all the way up through man. And there will be some interesting and important data actually showing that. And and what I like to do is I like to define aging as a gradual decline in cellular and tissue function. And that definition then tells me that there are going to be physiological, biochemical, molecular biological characteristics that will cause these this gradual decline in tissue function. And aging is determined by both genetic, by metabolic, and by environmental factors. And we'll be talking about those various factors and see how they interact and work in a combined fashion, so to speak. So I'm going to start by just sort of giving you this interesting analogy, and that is that indeed the molecular basis of aging and longevity, if we take a look at the life history of a racing car and the life history of a human, you almost see the same thing. The racing car was built to be very efficient and to carry out a very specific job, but it was not built to last eternally. It was not built to last, to never break down. And that indeed, after it has carried out its functions, it does show characteristics of breaking down. And what you do is you fix it, you take it to the garage and fix it, or you send it to the junkyard. And what we see in aging is essentially the same thing. We see the fertilization and the birth and development of a youngster, very efficient function, very healthy biological characteristics, reaches a very important peak, the reproductive age, where then there is a continuation of the species and generation, then reaches a post-reproductive stage where we do indeed experience the next generation. But at that post-reproductive stage, so to speak, what we're essentially seeing is the beginnings of the wear and tear and the development of the aging characteristics. And you either treat these with interventions or this results in death. And indeed, if you take a look at this series of they're all shown as babies, but if you take a look at the age at which intrinsic mortality occurs, you see a relatively high level of mortality during fetal development. Okay. But once that process is completed and you have the early postnatal age or the youth, you see that the rate at which death of that particular group is concerned goes down. And then as we mature and develop, the rate of death increases until we reach middle age and older. And one of the highest rates of death are at that particular point. This is all determined genetically. This is intrinsic mortality. And this then also is determined by the effects of the environment on your genetic makeup. And then there's a very small group that indeed get to be very old, oldest old, centenarians. And again, this is a major factor in the development into centenarians, is a genetic factor as well as an environmental factor. So in essence, those are the areas that I want to go into in more detail. And then that is what are the genetic factors, what are the environmental factors, and what is it about aging that occurs from yeast all the way up through man. And I will sort of discuss that with you. In fact, let's take a look at this very interesting phenomenon. We have here a colony of mice whose genetic background is very well known. Okay, and this is the theoretical survival curve of that particular mouse that has lived in exceptionally favorable conditions. And then if you take that mouse and you put him into suboptimal conditions, you can see that even though the genetics is there to live longer, they're going to die at a faster rate. That's telling us that the environmental factors can accelerate the aging processes. Okay, and if you keep that mouse in improved situation, in normal control situations, so to speak, then the lifespan of that mouse will show it and will increase. So indeed, that is a very, very important phenomenon. That is the phenomenon that the genes that develop, that determine aging and longevity interact with the environment to give you the final survival curve, so to speak, of aging and longevity. So, my main point and my first point then is that aging and longevity are under genetic control. And we know that, and we have now experimental data that indicate that yeast, nematodes, which are these small worms that have been actually a major organism that has made major breakthroughs in understanding the genetics of aging and longevity, as are fruit flies, rodents, and humans. Every one of these organisms exhibit characteristics that their longevity is under genetic control. And the genetic processes of aging are accelerated by environmental factors, and you can actually show this in these experimental animals. You can actually show that you can expose them to a variety of environmental factors, pollutants and toxins, and these pollutants and toxins will accelerate the development of these aging characteristics, so to speak. So, when the unicellular yeast ages, and yes, as I mentioned, the yeast cell does indeed age, they exhibit a decline in physiological function. And that decline is similar to the declines that we see in the aging tissues of higher organisms, including vertebrates. So what I want to do is show you the processes of aging that are actually programmed in the yeast cell. You start with a mother cell, so to speak, and that mother cell is going to give rise to a daughter. And that daughter will then go on and give rise to the first progeny. And the mother will then go on and develop and give rise to a second daughter, and then a third and fourth, and on down the line. If you'll notice as I drew this, the size of the mother gets larger and larger, an indication that she's beginning to show characteristics of aging, and finally, that mother cell will die. And that is programmed. And people, investigators have now used yeast to study major characteristics of aging. And we know that the characteristics that are developed in the yeast are similar to the characteristics that we develop in vertebrates. And that is, they experience sensitivity to oxidative stress and to oxidative metabolism. They experience sensitivity to toxic substances that accelerate their aging processes. So yes, indeed, the yeast does show characteristics of aging, and those characteristics are also found in higher organisms. So the age-associated declines in tissue function, then, we want to define them and try to understand them. And what do they involve? Well, one of them is cell proliferation. Cell proliferation is very important, and that's mainly because as our tissues and cells are damaged, they need to be replaced. They need to be replaced in order for that tissue to carry on its maximal function. And we defined aging as a gradual decline in tissue function. So aging indeed does affect cell replication. It slows down proliferation, and as a result of slowing down proliferation, we see that damaged cells linger longer in the tissue. And the more that these damaged cells remain and are not replaced in the tissue, then the function of that tissue declines. So this is essentially one of the factors that results in the gradual decline in tissue function. And this process of cell proliferation is a very important normal physiological process. You maintain the health of your cells and tissues by replacing the older ones and the damaged ones. That's a process called programmed cell death or apoptosis. And it is a process that is going on all the time, and you don't notice it because this particular type of cell death does not cause inflammation like necrosis does. This is a biological process of cell death that keeps going on all the time in the replacement of the damaged tissues in your organs, and you don't know that this is going on. But as we age, these processes slow down, damaged tissues stick around a little longer, and so cell proliferation is indeed an extremely important factor that determines the function of the tissue, and the function of the tissue is an extremely important factor in the efficiency of the function of that particular organism. Response to stress factors, an extremely important phenomenon. Stress is an age-accelerating phenomenon. By stress, I mean inflammation and infection and toxicity. These factors all accelerate aging. And indeed, studies that we've been doing in our laboratory now for the past 10 years indicate that there are major changes that occur in the biological processes and the biological mechanisms that regulate your ability to handle these stress factors, these inflammatory factors. And this involves that as we age, we develop a slight state of chronic stress. In other words, the biological characteristics that are occurring to indicate that you are reacting to a stress factor are endogenously increased. And we're walking around with that particular series of biological processes that can't be turned off, and that's one of the major problems. The problem is that you should be able to respond to an inflammation or a stress factor and turn it back off again. And our studies have shown that indeed we develop a state of chronic stress as we age, and our cells and tissues can't turn it off very efficiently. Now, the other very important thing is that in addition to the fact that we've developed an increased basal level of stress response, the second and important factor is that you can no longer respond to an external factor, stimulus. In other words, you have an increased level of a basal level of stress, but if you are then challenged by an external toxic factor, they don't respond very efficiently anymore. And so what we end up showing is that indeed, as we age, we become more and more susceptible to environmental factors. If our stress molecular biology, if our stress pathways, if our stress response is compromised in this way. Another very important system that is affected by aging is the immune system, the failure of the immune system. You're all familiar with the fact that we develop autoimmune diseases, and these autoimmune diseases are indeed age-associated, arthritis being one, and on down the line. There is a very interesting phenomenon that occurs as we age. The immune cells, the CD4 and CD8 cells, for example, in the younger animal, they are naive cells, and they are ready to receive the antigens that are going to stimulate the immune system. And then those immune cells become what we call the term slips my mind. The immune cells are then the naive cells versus the programmed cells, the cells that have actually been programmed to make antibodies. But as we age, those programmed cells don't turn over, they accumulate. And so the naive cells decrease in population, and the programmed cells increase. And so you have what we call an increase in the population of these cells that could indeed contribute to the development of autoimmune diseases. One of the areas of research that is extremely important for us here at UTMB because of our Pepper Center is the development of sarcopenia. For example, sarcopenia is the age-associated development of muscle weakness. I don't know of any studies that indicate that this is genetic. It is an aging phenomenon that I truly believe is going to be an environmental factor, and my own prejudice along these lines is that this is a problem that occurs with respect to the fact that cells don't turn over as rapidly, and as a result the myoneural junction, so to speak, may not be turning over as rapidly as it should to repair that particular junction, which is important for the maintenance of these particular muscles. Again, that's my particular explanation for that, and I don't have any experimental evidence for that, but I strongly believe that this is a cell-cell communication problem, which we will talk about a little later. Neurodegeneration is a major problem in aging. The nervous system is one of the most sensitive systems to aging. And what are some of the major factors that cause neurodegeneration or that cause the aging of the nervous system? Oxidative stress. Oxidative stress, which we will talk about in more detail later. What we actually know is that many of the neurodegenerative diseases are the product of damage being done to the proteins in the nervous system that cause these proteins to misfunction and that cause the development of neurodegenerative diseases. And I'll show you some very specific examples of that as we get into some of the examples of the molecular biology of these diseases. Okay, so the tissues of complex organisms age at different rates. In other words, tissues that have a higher oxidative metabolic function, like the brain, like the heart, and like muscle, for example, they develop aging characteristics much more rapidly than does a liver or a spleen or a tissue that also carries out oxidative metabolism but is not as intense a level of oxidative metabolism. So tissues do indeed develop at their aging characteristics at different rates. And there are now ways in which we can actually measure this, specifically with respect to oxidative metabolism and oxidative stress. These oxidation reactions actually damage macromolecules. They damage your DNA, they damage your proteins, and when they damage the DNA, they cause mutations, which can now be measured. When they damage your proteins, they cause modification of those proteins, and they can be measured. The molecular biology now of damaged proteins, which we called modified proteins or oxidatively damaged proteins, has now been very well worked out, and we can actually measure these proteins. And I'll show you some of this in a few moments, because when you actually then damage proteins that are extremely important for such functions as respiration, as protein synthesis, as cell replication, you then affect these biological functions, and these then are the functions that will result in the gradual decline in that particular tissue. So, cell proliferation is extremely important for tissue regeneration. You have to regenerate damaged tissue in order to function properly. In the young cells, in the young organism, this regeneration process goes on very efficiently. As we age, I personally think that there is a population of cells in every tissue that begin to age that are important in the regeneration of that tissue, and those are the stem cells. And I think that this is an extremely important area of research that we're going to see, and that is now actually emerging with respect to trying to understand what happens to aging tissue with respect to its regeneration. It is my belief that indeed stem cells may also go through a process of aging, which then compromises their ability to replace the damaged cells in a particular tissue. brain and neurons, for example, are extremely sensitive to some of the aging, oxidative stress and aging. And the skeletal muscle, as I mentioned, and the development of sarcopenia and others, the immune system and the autoimmune diseases. Aging of these biological processes depends upon the genetic makeup, but it is also the environmental factors that will affect these processes. So how do we know that aging and longevity are genetically determined? So let's look at that for now. We'll first look at the data that indicates that genetics is important, and then we'll also look at the data that indicates that the environment is also important. Well, first of all, as I mentioned earlier, one of the really most profitable and organisms that have been used to study aging in the past 10 years is the nematode. The nematode, the genetics, or the genome of the nematode has been completely sequenced and worked out. So we know the entire genome of this organism. In addition, we know that this organism is composed of 989 cells. And we know where every cell came from the time of fertilization on to the development of all of the various tissues. So it has turned out to be an extremely important organism to study major biological processes, including aging. And what it's been shown is that certain genes have been identified which, when mutated, or when their activities are specifically slowed down, these organisms live 2 to 3 times longer. So initially, their whole life cycle goes through in 1 month. You can get these organisms to live for 2 months and 3 months, an extremely significant extension of lifespan for these particular organisms. These genes have been identified, and I'm going to tell you in a few moments what these genes are, because you'll really be surprised what genes are the ones that extend longevity, because it turns out that these genes are also implicated in the extension of longevity in mice and most certainly in humans. So genes that regulate oxidative metabolism are associated with longevity. So genes that are associated with the regulation of carbohydrate and fatty acid metabolism, for example, are definitely associated with aging. And if you sort of mutate these organisms such that they develop resistance to oxidative stress and oxidative metabolism, these mutations usually lead to an extension of lifespan. And genes that are associated with resistance to stress, as I just mentioned, there are certain stress responses and stress pathways which, when slowed down, will also confer resistance to oxidative stress, and these genes are also associated with longevity. So similar mutations that extend the lifespan of these nematodes will also extend the lifespan of flies and mice. So is there, for example, an intrinsic or systemic control of aging? So what I'm trying to say here is that we have certain intrinsic biological processes that everybody experiences, so everybody is going to experience aging. I mean, there's no fountain of use here. And then there are the extrinsic factors, or the external factors, that will accelerate these intrinsic factors. And so the question is there an intrinsic or a systemic control of aging? And the intrinsic processes are those natural processes that occur in the absence of disease. That is, that no matter how healthy a life you live, there are certain intrinsic processes that are going to support the development of aging characteristics. And these are genetically determined processes. And they are, for example, the functions that are carried out by fat hormones, by growth factors, and by metabolites. For example, if we think about certain intrinsic properties, I'm going to talk to you a little later about the role of the energy-producing machinery, the mitochondria, in aging. Certain intrinsic characteristics in the mitochondrion, which are the electron transport chain, that transfer electrons down this chain that are important to make ATP, to produce energy. Okay, this is a very complex series of proteins that transport these oxidation reduction reactions that transfer the electrons to reduce oxygen, to produce water, and to produce ATP. Well, there is intrinsically a certain release of these electrons prior to their getting to the oxygen to produce water. When these electrons are released, they produce very, very devastating radicals. And these radicals then can damage the macromolecules like proteins. So these are some of the extrinsic factors, or excuse me, the naturally occurring factors, because this occurs throughout life. There's about a 1 or 2% release of electrons and mistransport of these electrons throughout life. And what happens is we get what we call that devastating cycle where the electrons are released, they damage the proteins. Those damaged proteins can't function properly. They release more electrons. So as we age, we increase this oxidative stress and the state of oxidative stress that we see in our tissues. And I believe that this is an important factor in the development of the gradual decline in tissue function, and that is the mitochondrial dysfunction. Other factors that are intrinsic involve the what we call genetic polymorphisms. There are certain characteristics of each one of the genes that we inherit. Some of these genes have produced proteins that have characteristics that carry out the same function, but they may be carrying them out a little slower, so to speak. An example of this is we know that some people will smoke heavily and will die by the time they're 50. And others will start smoking at the age of 10 and will still be smoking when they're 100, and they're not going to die at all. And that's a polymorphism that's been pretty well established. And that's a polymorphism that involves the genes that detoxify the smoke. For example, in the process of detoxification, you make these oxidative radicals that damage proteins. Well, the polymorphisms that give you a high level of detoxification you would think would be helpful. It's not, because it produces a lot of oxidative radicals. So people who have a high level of these enzymes are more susceptible to oxidative damage, whereas polymorphisms, where you change the structure of that gene slightly, those polymorphisms result in a lower activity of the enzyme, and as a result of that lower activity of that enzyme, you don't get as much oxidative stress and they don't get the oxidative damage, so to speak. So that's an example of an intrinsic factor that produces oxidative radicals, and indeed it's the same thing as being exposed to toxic substances that produce oxidative radicals that can do similar types of damage. So, physiological factors, then, and I will talk to you a little more in a few moments with respect to hormones and growth factors. So, the extrinsic factors, then, are the factors that actually will accelerate the aging process. Okay, so genes that extend lifespan in nematodes are also found in vertebrates, including humans. And these are the insulin, IGF-1, growth hormone family of genes. Indeed, it is the insulin pathway, the insulin IGF-1 growth hormone pathway, the signaling pathway that plays a major factor in determining longevity. If you will remember, insulin regulates carbohydrate metabolism and oxidative metabolism. And in these nematodes, one of the major breakthroughs that came was that there was a mutation of the insulin receptor that resulted not in knocking out the receptor, but what we call a reduction of function mutation. What that means is you mutated the insulin receptor in such a way that could carry out its function, but at a much slower rate. And by mutating the insulin receptor and producing a reduction of function of the insulin signals that go to the metabolic genes, to the oxidative metabolism genes, so to speak, you reduce that entire function, you increase lifespan. And this is now very well established. And you can do this in flies, you can do it in mice, and I'm sure that this could also happen in higher organisms. So these genes regulate oxidative metabolism, and they control longevity genetically by reducing the function of that particular receptor so that it reduces the signaling pathway to oxidative metabolism. So genetic longevity is of such biological importance that indeed it has been conserved throughout the evolutionary scale. Okay. And what I'm saying is as follows, and I'm going to show you this in a few moments, that the insulin pathway has been shown to be of importance in regulating the metabolic processes that regulate longevity. And we see that same pathway in nematodes, Drosophila, mice, and humans. The same biochemical reactions conserve or extend lifespan if you slow them down. Okay, so what this means is that the process of longevity, the process of lifespan, is so important that it has been conserved throughout evolution. Now, this is a little animal that we work with in our laboratory, and this is a mouse that has a mutation that results in a reduction of the cells in the anterior pituitary that make growth hormone. As a result of the reduction or growth hormone deficiency in these animals, you get a reduction of circulating insulin and IGF-1. So we're essentially mimicking what we see in the nematode and in the fly by reducing the amount of insulin or circulating insulin, which is the same way as decreasing the insulin pathway. Remember, we cannot mutate the insulin pathway in higher organisms. We'll kill them. They'll develop diabetes right away. But what you can do is reduce the insulin signaling pathway by decreasing the amount of circulating insulin. So the reduction of growth hormone results in reducing the amount of insulin that's produced and the amount of IGF-1 that's produced. And our studies in the past few years have shown that indeed that very same pathway that decreases in the nematodes and in the Drosophila is also decreased in this mouse. And this mouse lives longer. And so we believe that indeed the insulin IGF pathway plays an extremely important role in development, in determination of longevity and aging, and that's mainly, we think, because it targets and regulates oxidative metabolism and carbohydrate metabolism and slows it down. And here you can see a survival curve. This is the normal wild-type mouse, and this is the mutant mouse, and they live anywhere between 40 and 60% longer than normal, which is quite an increase in lifespan. A normal mouse will die by the time it's 24 or 25 years, and they're very old when they're 30 months. These guys will go up to 45 to 50 months. And so it's a very, very a significant extension of lifespan. Oh, okay. So now, here's what I want to show you with respect to the evolution. Here we have our mouse. Our mouse has the insulin IGF-1 pathway, right here. And insulin receptor now goes on and sends signals to the proteins that are going to activate what we call these fork-head transcription factors. These are the proteins that go from the cytoplasm to the nucleus to activate the carbohydrate metabolism and oxidative metabolism genes. Okay? So this is a well-established pathway of how insulin actually conveys its ability to regulate carbohydrate metabolism. And this is the worm. And the worm has the same thing. DAF2 in the worm is the same as insulin receptor in the mouse. Okay, age 1 in the worm is the same as PI3K in the mouse. AKT is, again, these are proteins that transmit signals finally to the genes that are going to activate the metabolism. So the same pathway in the worm as in the mouse is associated with determination of longevity, because if you slow this pathway down, you're going to slow down oxidative metabolism. And we see the same thing in the fly. the same response. You mutate or slow down the insulin pathway in the fly, you will get an increase in longevity and a decrease in the stress response. And here we see yeast, and again, you have now mutations in yeast that slow down glucose metabolism, and they extend the lifespan of yeast. So this is a phenomenon that is so important that it has been conserved through evolution. So is human aging genetically determined? And we have lots of evidence from this, from some of the genetic diseases that we're pretty much familiar with. Acceleration of aging is genetically determined, and there are two diseases that we're familiar with that show this. One of them is Progeria, and the other one is Vernors syndrome. Now, Progeria is a disease that accelerates aging 8-fold, so that these kids usually don't live beyond the age of 12 to 14. And when they reach the age of 12 to 14, they look like they're 80 years old. And I'm going to show you this in a few moments. In the past year and a half, the genetic basis for progeria was not known, and it's a very rare disease. In fact, when I moved here back in the early 1980s, there was a progeria family in Beaumont, and I don't think that anyone ever really got to study that family, but in any case, it's a very rare disease. There are only about 40 or 50 of these cases throughout the world, so to speak. But the cells from this mutation have been cultured, and you could now go in and look at and identify the gene. The gene has been identified, and it's called lamin A. And lamin A is a gene in the nucleus that conserves the structure of the nucleus, so that the nucleus of these children shows blebs and shows all sorts of faulty transcription of particular genes. Vernors syndrome is another one that's been known for a long time, and the mutation there is called the REC-Q helicase. And in this case, this particular syndrome involves the regulation of DNA synthesis, cell replication, the regulation of DNA repair, and the regulation of transcription of genes. So these single mutations, so to speak, have global effects upon the genome to produce the complexities that we see in the development of the characteristics of aging in these mutants. So the longevity is also genetically determined. And this is based mainly on the fact that we now have very good documentation of centenarians. And we also have documentation that certain families are resistant to certain diseases, so to speak. And we also have the polymorphisms that I just mentioned with respect to the smoking. But here's a picture of one of the progeria children. Okay, so it's a disease that he's saying it's a disease that makes my body get older 8 times faster than ordinary people. It's a very rare one, and there are just 4 kids in the United Kingdom and only about 40 around the world. And the average lifespan of these children is about 13. And there is no cure for this, but you can actually see that this one mutation in the lamin A gene results in developing the complexities that we see in these multiple characteristics of aging. They look like they're 80 years old, and they're only 12 to 14 years old. And here are a couple of the other kids that have progeria, so to speak. Okay, now, this is a picture of what the laminin gene looks like in your genome. And what happens here is that the messenger RNA that will make laminin is going to come from this particular region of the DNA, and this is the messenger RNA. So what happens in the progeria? What happens is, you see that dark spot right there? There's 150 nucleotides that belong in there that have been deleted. It's a mutation. It's a mutation that deletes that part of the protein so that the protein is abnormal and it's being synthesized, but it can't carry out its functions completely. And as a result of not being able to carry out its functions, it now develops the characteristics of progeria, which are indeed multiple genetic characteristics. So this particular gene has a major effect on the way many other genes are expressed. Now we go to Vernors syndrome. This is George Martin, an investigator who has devoted much of his life trying to understand Vernors syndrome. And he has done many cohort studies. And this is a German, he studied one of the German and one of the Indian cohorts. And this guy's 36 years old. And he looks like he could be 70 or 80. And this guy also is 50 years old. And so you see this mutation, which is a mutation that affects the helicase. And the helicases are genes that open up the DNA when the time comes for them to replicate and to transcribe particular genes as well as to replicate the DNA. So in essence, these guys also show characteristics of accelerated aging. And it's really interesting that they show many, many characteristics that you would see in a truly wild-type 80-year-old. And they develop these characteristics by the time they are young adults. Here is a Japanese lady, for example, who had Vernors syndrome. This is one of the classical pictures. And she displayed defects in the DNA replication, in recombination, in repair, and transcription. And she is a teenager here and 48 here. Okay, and she had 8 children, and she lived to be 57 years old, which is a pretty long life for a Vernors syndrome. And here's another Vernors syndrome showing the facial characteristics and the leg characteristics of aging. And finally, I want to go the other way. So we have genetic data that accelerates aging. We also have examples who we could predict and say, well, their genes were really centenarian genes. You know who this guy is. It's Bob Hope. And Bob Hope lived to be 100. Okay, and we don't know about Bob Hope's genetic makeup, but we know one thing that's very important that I think that Jim mentioned to you. He kept busy, he was happy at his work, and he loved what he was doing.